But even for French people, it's not easy, so no worry.
Use the pronunciation.
We are going to try. Quiterie de Beauregard.
Wow, you're doing well, you're doing well. No, but the QU for English-speaking people is just QI, so it makes sense. Et c'est parfait. Thank you. So I'm very happy to be here today and grateful also to see that so many people are working for long-acting injectable drugs development. If you remember all, one patient on two doesn't take its treatment as prescribed. That's a figure from WHO. It also means that it has been prescribed. So we have a lot of hope for long-acting injectables in many, many indications. And so it will be a bit more exotic now because we'll speak about malaria and in particular malaria vector control. So at Medincell, it's actually, it was mentioned yesterday, we work also on some drug that have approval in the veterinary drugs, but this ivermectin is also approved in many neglected tropical diseases. So it has been used in millions of patients worldwide and in low and middle income countries in particular. So the most known indications are river blindness and also lymphatic filariasis and also other entities. So what is very important with ivermectin is that the safety is very well known. That's a big topic with long-acting injectable, and it's also a chance to have a molecule with such a track record on the safety. So the approach, maybe some of you have heard about this, is to use ivermectin to control malaria transmission via its vector control. So the principle is that people will receive an injection of ivermectin, long-acting, to cover the all rainy seasons. So it's mainly in a very high transmission setting. And the mosquitoes that will bite people will get the effect of ivermectin, meaning either they will die or they will lose the fertility. So at the end, we expect to have an impact on mosquito density as well as mosquito density, and so being able to decrease malaria transmission as a consequences. So to date, the project is still has completed successfully the preclinical stage, thanks to a consortium made of people and colleagues in Burkina Faso, IRD, IRSS, and CIRDES. We could make some PK, and I will present you the result just after, but at least it's working, which is a good news. We also had some positive feedback for the clinical phase 1 from MHRA. So the design of the phase 1 will be very simple, a classical ascending dose. We also have the chance to have Medicine's patent pool supporting us in order to find some partners, in particular for the manufacturing. It's a challenge. We can discuss that because, as you may imagine, it's not a product with a high commercial potential. But nevertheless, we are confident in the clinical results, and we are currently preparing the phase 1 clinical trial that should start next year, hopefully. So as you will see on this graph, we had in the first pharmacokinetic study that we run, the achieve of 3 months expected concentration of ivermectin in cattle. This was done in cattle, and this is already a very good news because this is what expected, at least to have this 3 months duration. So it's a bit, for those who are not used to this kind of study, so basically the efficacy, even for human, will be measured by the mosquitocidal efficacy. So it means that basically you count the number of mosquitoes which die, and when you put all this in a modeling, again, in a modeling, it gives you prediction of what can be achieved in terms of decrease in malaria transmission. So this study was run in Burkina Faso on 2 sites, one with multi-resistant, mosquitoes to insecticides, and the other one in Diebougou in high diversity species, because these are the main criteria that can make the efficacy vary. In cattle, again, and so with 2 arms, one arm received a single injection of ivermectin, and the other one, nothing. So as you can see in the graph, in the upper graph, the bioefficacy was achieved. We had a daily survival that wouldn't exceed 5 days during at least 4 months, and the hazard ratio was superior to 4, which is again what was said by WHO and what was achieved with this formulation. In terms of mosquito species, we also could observe the expected efficacy on the most relevant species in Diebougou. So again, in this case, this was the target product profile was achieved for this formulation. Importantly, because you may ask yourself why, would I get an injection of this drug to protect other people and the community from malaria transmission without any benefit? I think there are not a lot of benchmark, but obviously it's very, very important to have user acceptability feedback on this, as it has been discussed a lot in this conference. So we started to do it among donors and also implementers, because we thought that it would be better to wait a bit to have some data to do it with patients. The good news is that we had quite interesting feedback also, because I think that the need is huge. As you may know, the malaria, especially in high burden to high impact country, is not decreasing. So there is definitely a need for additional tool, complementary tool to vaccines, SMC, and also treated nets. So I think this is why. And the idea, we are aware that to ensure the success of this program, the idea will be to pair it with other intervention, SMCs in particular. And I invite you to look at the work that Medicines for Malaria Venture is doing on long-acting injectable drug for seasonal malaria chemoprevention, because this is highly, highly needed. I think that's all. Sorry. With that, I thank you, and again, I'm available for any question that may come from this talk.